My name is Dalip Sethi. I'm from Terumo Blood and Cell Technologies. This morning was very exciting with Dr. Koya's presentation. He shared the novel 3-in-1 data in quantum flex. My key highlights, and that's why I have my notepad with me, he opened the presentation showing the flexibility of the quantum system to grow different type of cells, suspension cells, adherent cell. This was very exciting to see and how he has got his protocol to work in quantum flex system. Then he highlighted his protocol where he used quantum flex in conjugation with Fujifilm Media Prime XV, expanding TCR-T cells. It was very exciting to see that he could grow one of the donors, donor 2, up to 11 billion cells, starting with a seed of 10 million PBMCs. That was very exciting. He got two different donors showing data on different donors, but that was very exciting to see with a small bioreactor. We have a standard size bioreactor too, but he could go up to that many cells in just 10 days. So very exciting. And also he highlighted the ease of use, that you can sample the cells and the metabolites using the sampling methodology that we have in the quantum flex system to do the immunophenotyping and all kind of analysis, cell analysis that you like to do when you're expanding cells. So that's very exciting. Overall, how he summarized that it's a very easy to use system and you can develop your protocols, which was exciting again. The novel 3-in-1 data and all the TCRT data that he showed today was super, super exciting, along with all the phenotyping and all the potency assays that he showed. Going back to the presentation, one of the things that he highlighted is how you can use the system in a GMP compliant way as a closed system. So as you think about unit operations, when you think about the activation, the transduction, and the expansion, if you're using manual systems where open steps are incorporated, it's very hard to optimize that for commercial manufacturing. And you always have that risk of contamination, the risk of losing your batch. So he highlighted, I'm just repeating his words, I say those words all the time, having those automated closed systems will help the researchers definitely, first of all, optimizing their protocols, but also thinking about as the next phase of clinical trial, as they head towards the commercialization, having a system that allows you reporting, monitoring your systems, and also easy to scale out.